Genetics and Precision Health. This is going to be moderated by Marg Spoelstra, who will be introducing herself and the panel. They can give me a bio for you.
That's good.
Get to make up your own stuff.
I get to tell people what I want. Yeah, I know. That's a dangerous thing to do. Whether you want to hear it or not, that's right. I'm Marg Spoelstra. I'm the CEO of Autism Ontario. And I have been delighted to be part of POND since its very beginning. And every time I come to these meetings, what thrills me is to see the people who have been participants right from the beginning or for many other years. I lean in, that's what I'm supposed to do. And also to hear about the exciting work that's going forward and how things have changed even in the time that we first started. So to be part of this and to be part of the change is really exciting. So thank you all for being here. And so now it's time for us to listen to more of our esteemed panel experts today. And we're going to have each of them speak for about 10 minutes. And we're not going to take questions after each presentation so that everybody can get through their presentation. But write your questions down, and then we'll see what we have time for. Okay. So it's my privilege to introduce you to the people who are here today. Mukti Reddy, right here on my left. She is the chair of the board and president of CARE Ash1L. And this is looking at research into understanding the molecular mechanism of the Ash1L. I'm saying that right? Ash1L. And to expedite the development of therapeutics for Ash1L gene disorder. Mukti lives in Toronto and has a son with Ash1L mutation. And then to her left is Dr. Danielle Baribeau, who is a child and adolescent psychiatrist with a focus in autism, rare genetic neurodevelopmental disorders, and psychopharmacology. As a clinician scientist at Bloorview Research Institute, she leads research focused on understanding and supporting mental health in neurodevelopmental conditions. Dr. Baribeau lives a busy life with her 3 young children, and in her spare time, you can find her attempting to garden unsuccessfully and hanging out at playgrounds in the West End of Toronto. So you'll be hearing from her as well. And then to her left is Dr. Stephen Scherer, who is the Chief of Research at the Hospital for Sick Children and the University of Toronto McLaughlin Centre. Recently, Dr. Scherer was awarded, I'm going to say this, I'm not sure, De Bresson Award. Did I say that right? Not bad? Okay. For molecular medicine for his contributions to the discovery of genome-wide copy number variation, CNVs, and its role in autism and other neurodevelopmental conditions. Dr. Scherer grew up in Windsor and was a competitive hockey and baseball player. Now he enjoys gardening and spends time with his family fishing and kayaking up north. So it is my pleasure to introduce this lovely panel to you. And I believe we're starting with Mukti. Is that right? Okay, come on up. Yeah. Okay, I think it's right there. Let's click on here. There's yours right there.
Thank you.
And then you just have to click as people move through.
Thank you so much. Good morning, everyone. Thank you very much for the introduction, Marg. My name is Mukti Reddy, and I am the co-founder and president of a Canadian charity called Care for Ash1L. Before I start, I would like to thank POND for giving me this wonderful opportunity to share my journey and my son's story. So we all have a reason here to be today, and my reason is right here on the screen. It's my son, and his name is Karthik. He's 11 years old, and a lot of our Ash1L warriors. I'm here today not only as a parent, but also as a relentless seeker for answers. My journey into the world of genetics and neurodevelopmental advocacy began because of my son. He made me question everything that I was told and fight for everything that he deserves. What actually started as a quest or a support for research has now become a compelling factor or an evolving foundation for my charity Care for Ash1L, which connects Ash1L families across the continents. And today, I'm here to share how a single diagnosis through PON study has transformed my family's life and also sparked a mission. Just a bit of a background and history about my son. He was born in India at 36 weeks in September 2013 through C-section. Everything was normal. But when he was 6 months old, he developed chronic constipation that required daily medications, oral medications, and enemas. When he was about 15 months old, they diagnosed him with severe D3 deficiency, and the doctors thought that could be the reason behind his delayed milestones. Nonetheless, he always had delayed milestones, like he didn't walk until he was 18 months. He had no speech for about 4.5 years. Now, coupled with low muscle tone, he currently struggles with a lot of dental issues. He had a couple of surgeries, with the latest one being labial and lingual frenectomy. He was recently diagnosed with molar incisor hypomineralization. I hope I said that correctly, which means his molars are almost like sponge. And unfortunately, the condition also spread to his other teeth. He's under constant observation under the supervision of dentists. He's not allowed to eat anything that's sour or has a lot of citric acid in it. Again, he was also diagnosed with orofacial myofunctional disability, which in common terms would mean he cannot use or sense a lot of muscles around his cheek or his cheekbones. So I guess all of these coupled with this, he doesn't have a fully developed sense of smell and taste. He cannot chew his food, often causing vomiting and gagging. We did pursue a lot of non-invasive biomedical treatments, and we still continue to do a lot of OT and speech therapies, but we don't see a lot of improvement in him. He was diagnosed with autism here in Ontario in 2017. We accepted the diagnosis and the label, but that did not explain everything for us. So we enrolled in the PON study in 2018. We did the whole genome sequencing. And in 2020, we got the report that he was, he has a deletion in his ASH1L gene, which is a gene in chromosome 1. And that is one of the leading causes for neurodevelopmental disorders. So that was when corroborated all his health issues that we saw in him. And for the first time, our confusion had a name and our path now had a direction. Now, ASH1L mutation is considered to be an ultra-rare genetic condition. There's very little information or resources available online. We have very limited clinical guidance also for it. So just like everybody else does, as a mom, I was desperate for answers. And I delved into the vast ocean of Google. And I was very lucky to find another mom. Her name is Kimberly. I found a post of her on a Simon Searchlight Facebook group. And surprisingly, she's also from Canada. She's from BC. And we connected. And we realized there was a lot of things common between her son and my son. And we started having long conversations. And it was very comforting because I didn't have to explain anything. There was a lot of validation, something that I was missing for. And through those conversations, we thought, or we realized that we should have a dedicated group for ASH1L families. And that's when we created Care for ASH1L. It started with just the two of us, but at least we knew that we were not alone in this journey anymore. So we registered Care for ASH1L as a charity, as a Canadian charity federally in 2021. It exists to support, connect, and empower the ASH1L families through advocacy, research, and capacity building. So in our Facebook, we currently have about 200 members in our group. And all of our conversations are very real, raw, and authentic. It's a very crucial forum for us because we tend to share a lot of our journeys, like our kids' odysseys, their challenges, their clinical manifestations, their complexities, their medical complexities, school advocacy, everything. And it's like a moment of catharsis for a lot of parents, where we share everything that we can without any inhibitions and prejudice. We often tend to have video conference where families connect. I would like to say that we don't offer any medical advice, but because of the limited clinical guidance, we tend to support each other, by saying what drugs have worked, what may not have. And oftentimes it then helps empower the families to reconnect with their physicians, to get better diagnostics, better treatment plans. I would like to say that, this is not just a support group. It's a living library of hope with lived experiences. And that's what we try to do. We try to, it's like an establishment where we coexist with our physicians. It's a symbiotic relationship that we share. Our next step is to have a similar family day with our Ashwanal families. I hope we succeed. And I only have probably 2 more minutes, so I don't want to take much of the time. I would like to end by saying that everything that I do is for Kautik and for other kids like him. It's what my journey started as a diagnosis through porn study, and now it has become a beacon of hope. And with that hope, I want to share something very timeless, and that is, it doesn't take a lot to be deeply and profoundly human. And and when you have children like Kautik, it gives our mankind the rare opportunity to showcase their human spirit. And in that spirit does not just rely on being a perfectionist, but standing up for those who matter the most to us. Thank you.
Thank you so much, Mukti. That was really beautiful. Yeah, just have to catch my breath and think about your words. Thank you so much. Okay, I'd like to invite Steve Scherer up at this point.
Okay. This was a spectacular presentation we just heard. Thank you so much for sharing your story. And I'll make reference to it in the next few slides that I have. Okay, so this is a slide that many of you have seen before. For those of you who are new, I show this at pretty much every talk that I give. And my comments today on the slide are for autism spectrum disorder, but they relate to any of the neurodevelopmental conditions that we study here in Pond. So why do we perform the type of research you heard just beautifully in the last talk, and also from Dr. Anaganastu's opening presentation? But I think most importantly, it's important to point out that the truth will be found in science. Everything we're hearing south of the border these days, alternate explanations, pseudoscience, the main advances in biomedicine, not the main advances, all of the advances in biomedicine over the last 100 years have come from scientific studies happening at institutions like that we have here at Sick Kids and Holland Blue Review and part of this Pond partnership. And I wanted to really thank the Ontario Brain Institute for supporting fundamental scientific research. So thank you, Jordan, and please pass that back to the rest of the group. The second point is earlier detection of neurodevelopmental conditions and can benefit health outcomes. We'll hear lots about that from Danielle. Third, better understanding of the causes of autism or NDDs will benefit medical management. And we heard that in the last presentation. Fourth, due to the heterogeneity involved, individualized approaches may be the best way forward. And you're going to hear much about this. And as we just heard, understanding these very important human developmental conditions, we understand those features that are most important that make us human, things like cognition, speech, and behavior. And by studying people who have these conditions, we actually understand ourselves much better. And this is so important in the understanding of biology, physiology, medicine, but in sociology also. These are just a few other examples of people who have received their diagnosis as part of the study over the last few years. And there are many more, as you'll see in a moment. So the idea behind precision medicine, as Advocacy laid out, and I think Danielle will also talk about this, is to identify the specific forms in as much as we can of the different conditions and all of the medical attributes, and then try to treat them with the best intervention that's possible for that clinical presentation. That's what precision medicine or personalized medicine is. You've seen this slide before, some of you. We've been doing this study now, the whole genome sequencing study, for a better part of almost 15 years now. And we're very proud that we've collected and studied over 4,000 families from across Canada. 2,625 of these are part of the POND study. This is for the autism, but we've also now started to look at other neurodevelopmental disorders, and we've now done sequencing of roughly 500 or so other families. And there's many, many more in the queue. And as Evdokia pointed out, we now have funding to actually do the sequencing. So this is so exciting for us. And this is the distribution based on postal code of the participants right across Canada. So elbows up, this is a Canada First project. It's the biggest sequencing project in Canada and one of the biggest in the world. And I really have to acknowledge the families who are participating, in other words, you, for helping us to really implement this concept of whole genome sequencing and precision medicine. Almost all of the projects in the world are based on what we've done in the earlier autism studies. So congratulations to you. When we have your genetic sequences, we can actually group them based on the characteristics. And as Evdokia pointed out, we have very good representation from the Ontario population, as shown on the bottom right in this slide. But of course, as she pointed out, there are reasons why we want to overrepresent certain groups to increase sample sizes to do informed statistical analyses. Okay. She mentioned the Precision Child Health Initiative. There's actually a project led out of, well, so there's the POND project that Evdokia just touched on very quickly. And then I'm leading an equivalent project out of Sick Kids here, where we'll also be doing 10,000 participants. And then there's other projects. There's 12 of them going across Canada, and you'll see some names. Jen Crosby's name is on there, and others. And this will be to develop a resource of 100,000 participants. There was a project like this in England a while back called Genome England, and one in the United States. So this is something we've been striving for, and I just wanted to emphasize again that it's really built on the foundations of what we've done here, participants in this room. So congratulations to all of you. This is a success for your efforts too. I don't have a lot of time to talk about this, but as I said, this is going to provide sequencing, the dollars to do the sequencing. 50% or 50,000 of the genome sequences will be done on the 13th floor in the lab that we'll tour a little bit later. And the other 50,000 are actually being done in two other centers, one in British Columbia and one in McGill, and they're tied together in a national grant from the Canada Foundation for Innovation, and I'm the lead investigator in that grant. So in fact, we'll have kind of oversight on all of the sequences. The nice thing here is that there's comfort in numbers too. So the Autism Project now, we've made that data available on the cloud, and there's over 342 investigators from 65 institutions in 20 countries using the genome sequence and the clinical, the phenotypic information to ask specific queries about, why autism came about, how we can adopt precision medicine approaches, anything really related to understanding the truth or the science behind, in this case, autism. I did want to point out that we also have companies involved. As Evdokia said, we think that they're going to ask very informed questions and bring new resources and new ideas to the table. So we want everybody who has a smart question to actually have access to this data, and we consent for that up front. This is my slide I made specifically for you for today. So we know autism and many of these neurodevelopmental conditions have a significant genetic component. We know that. We've known that for a long time. So for Robert F. Kennedy Jr., in case you missed the news, there's already 100 genes that have been identified that are used in a diagnostic setting for autism. And these are tested for now around the world. So in a way, we know what quote unquote causes autism. We don't have all of the answers, but we have a very good starting point. And much of that maps back to this project. So you should be very proud. I'd really hope to actually have a new slide showing the Maple Leafs but we're going to go with Edmonton, as Jordan said, and they're going to win this year. So, and we published a really important paper just a few months ago, and you'll see authorship from many of us here, where we actually this was the first 100 families from the missing study, which is largely supported by Pond, that received their genetic information. And I think Coutique's data is in there. You can find, this is just the first table from the first 100 samples. There's another table on the next page I don't have time to show you. And many others of you in the room, where we share with the medical community and anyone who accesses this paper, the experience of what the impact was when they received their data. Okay, so it took a long time, and we had the parent advisory group involved, the patient advisory group, actually had input on this data, and I think it's based on the experience of the genetic counselors interacting with all of you, getting your data back. And we're happy to share this with you if you haven't seen it. I think Danielle is going to show this slide. My guess is it's from a review that her and Jakob Vorsman wrote, and around the novel therapeutic targets. So of these 100 genes or so, these listed on the left, and ASH1L is not on there, but it would certainly be one of our top genes, as you heard. They map into biological pathways. When Evdokia talks about biology, these are the cellular pathways that these gene products or proteins interact in. And she kind of skipped over one slide, but yeah, you can see this here. This is our baclofen clinical trial she's studying. That drug would actually try to interact with this genetic pathway here at the GABA-R pathway. So depending on the genetic alteration you have, the idea behind precision medicine, and you would try to use a small molecule or a gene therapy-based approach to modulate that pathway specifically. And one of the challenges in clinical trials now is we're not going for specificity yet. That's going to come in the next round. We test all individuals that have a specific diagnosis. Okay, so I think the data is only going to get better and better as we have more specific populations that we're testing in. And I just have two more slides very quickly. This is a slide we put together for precision medicine review. So many of these neurodevelopmental conditions are rare genetic disorders. And as I said, for autism, we have over 100 different forms, if you will. There's actually 10,000 different rare genetic diseases in the human population that have been documented. And this is based on a review we wrote between Sick Kids, Great Ormond Street Hospital in London, the Children's Hospital in Melbourne, and also Boston Children's. And what I wanted to point out here is that I can't see very well, and 30% of these different conditions, they have other clinical symptoms that overlap with, thank you, with other disorders that have different names in the clinic. So often when we get a genetic diagnosis or we understand the pathway, there may be clues there to use or borrow treatments from other conditions and use those in the autism or the OCD or the ADHD cohorts. And believe it or not, when we did a literature survey, 12% of the disease-associated genes actually have interventions attached to them in some way. So again, we're trying to cull all of the information on patients in medical records databases from all the medical literature. So when we get a genetic diagnosis, we can have an informed path forward on how that individual should be treated. And then lastly, I'm not going to go into any detail here, but the advances in technology have just been astounding, where there's new gene-based approaches that have been developed. And I think we're going to get a question later. And I wanted to just point out John Oldenhoff, who's in the audience here, who's been part of our study for a long time. His group has developed a gene replacement approach for the STXBP1 gene. And I'm going to try to call you out maybe a little bit later during the question, John. But it was based on first finding the genetic finding, understanding the pathway, and then using one of these advanced gene therapy-based approaches to try to modulate or correct that alteration. We can do this at the DNA level using technologies called CRISPR, which you've probably, many of you read about. At the RNA level, now we can modulate using DNA sequence changes, alterations, or engineering, we call this, protein replacement, even modulating circuits using neurostimulation and neuromodulation approaches. The key is to understand what form of the NDD that individual has and what treatment may be the best path forward. But it's still, this is all still in the realm of research. And that's why we need to have the feedback from all of you through meetings like this today. I think that's my last slide. Thank you.
So hi, everyone. I'm Danielle. I'm going to attempt to bridge the two previous talks. So a little bit on family perspective and research, and I'll try to pull those together about what's happening clinically now and where are we heading when it comes to genomics. So my plan is to talk about testing, then about clinical care, and then our future efforts and projects that are underway. When we think about clinical investigations in genetics for kids with neurodevelopmental differences, it's important to separate rare and common. So of course, genetics play a factor when it comes to how brains develop, right? If you have a parent or a grandparent who has a lot of ADHD traits, there's a good chance you will have a lot of ADHD traits. These are, if you think about kind of common differences in the population, we call these common genetic differences. They are there and they are important when it comes to shaping brain development and likelihood of a diagnosis. We can test for these common differences in big cohorts in a research setting. We are not yet using this clinically. So if sometimes we'll hear, you know, is this single SNP associated with, or can I do the autism polygenic test? It's not something that doctors do in the clinic. This is a research thing. What we do in the clinic is look for these rare and high impact genetic differences that you don't find commonly in the population. And so much of the clinical translation of genomics at this stage is in this rare space. This graph I took from an old paper of Steve's with knee, where it's this ball and cup model, which some people like or don't like. But the idea is if you have a cup and you fill your factors contributing to a diagnosis to the point that it tips over that dotted line, that's when you get a diagnosis of something. And the factors contributing can be red dots, environment, can be green dots, these weak common genetic factors. And those we really assess through talking to patients and families. What we can test for are these strong big blue dots, okay? So that's the first step. The challenge is when we look at rare genetic conditions, like we heard about ASH1L, in intellectual disability, probably somewhere between 30 to 50% of people will have a rare genetic underpinning. In autism, it's a bit lower even more so for ADHD, OCD. But when we look, 80% of the conditions identified are ultra rare. And what ultra rare means is when you go to your doctor, they'll have no idea what this is. They'll never have heard of it. They may be embarrassed about that, so have some compassion for them because they'll think they missed the lecture on it. They didn't, they're rare. You may be the only one or one of a handful in the country with a kid with this condition or affected by this condition. And there are huge gaps when it comes to what that means and what to do about it. This graph is from a sick kids cohort of children with medical complexity. So 90% of them have brain or neurologic impact, but also medical complexity. And of them, the biggest bar there, let's see if I can do it. Yeah, this biggest bar here is showing of the 700 kids in the cohort, 200 were the only ones in that cohort with that condition. And so this is really tricky when it comes to trials and generating clinical care because they're that rare. Common, but individually rare. I looked at the POND genomic data recently, and these are conditions that are present in more than 5 individuals in POND, of those have been sequenced. Black are not rare, so Down syndrome is not a rare condition, in fact. Blue are rare, so less than 1 in 4,000. And then light blue here are the ultra rare, less than 1 in 50,000. So if you're in the rare group, you may have a research cohort, you may have a trial emerging, you may have a review article that tells your doctor what to do. The ultra rare space, that's much less likely to have occurred and is a gap that we need to address. We surveyed it recently. So we did a literature review of 100 newly described ultra rare genetic conditions. Many of these were part of this network or through collaborations through this network. And in the initial descriptions of them, so the publications, understandably, they're very much focused on the biology. What are the sequences? What did the cells look like? What happens to the proteins? And there's some description of phenotype. So what do these kids have or not have? Mostly they're little kids, like 2- and 3-year-olds. And usually the descriptions of most of them, if you see this column, are superficial or completely absent. So not a lot of information about development, not a lot of information about mental health, which is a pet peeve of mine. Most of them will say X number of children had behavior problems. That means nothing to anybody, frankly, sorry. Feeding and growth challenges, what treatments worked, pain, sleep, and quality of life. This is not a criticism of the initial publications, but a gap that we need to address. And this collaboration we need with clinicians and basic scientists when we describe these conditions. I'll give you now some examples of how a genetic diagnosis can inform clinical care now, and then I'll tell you about where we're moving. I'm a psychiatrist, this is a psychiatry example, and I'm gonna focus on depression. So we know children with neurodevelopmental conditions are more vulnerable to mental health outcomes. This is from POND data where we showed, looking at, this is a depression score, anything over about 60 or 65 is elevated compared to the general population. And we showed about 1/3 of autistic kids in POND had elevated depressive symptoms when they were about 10, 11 years old, and much of them persisted. And so this is not, clinical diagnosis of depression, but depressive symptom. It shows what a lot of us see is that adolescence is a really vulnerable time for kids with neurodevelopmental differences, and it may be hard to treat or get the supports you need. Unlike with grown-ups who show up to clinic and say, I'm depressed, help me, 11-year-olds usually don't, 12-year-olds usually don't. And so it can be really tricky for families and clinicians to figure out, is this kid depressed? Is he going through puberty? Is he sick? Is he doesn't like his class? Like what is this behavior, which can be social withdrawal, loss of interest in activities, sleep challenges, like what is going on here? Some of our rare genetic conditions have a known association with some serious mental health outcomes. So 22q deletion syndrome, for example, we know about 1/4 of kids will get psychosis or psychotic disorders. So if I see a young person with 22q deletion, and I'm thinking, is this depression? I am also really thinking about psychosis, risk factors for psychosis. Don't you dare smoke weed is the next conversation we have, and counseling families around what to watch for. So in present day, for a rare genetic diagnosis, we can start to anticipate some and watch for things that may be coming down the road to get a little bit of an early heads up about what might be happening there. I pulled a few other examples of how it's affected my practice. One would be these late onset regression. So we know about regression in skills in little kids with autism diagnosis or other developmental presentations, but this can happen to teenagers and young adults too. It's less talked about and it's less well understood. In certain genetic syndromes, presence of a regression or a loss of skills can be associated with a certain phenomena and direct us to treatment. So kids with EHMT1 or Kleefstra syndrome, there are case report level data showing in adolescence, these kids will have a period of insomnia or may have a period of insomnia 2 to 3 days associated with skill loss. And if we can really aggressively treat that insomnia with some pretty high dose intense meds, it can stop that from happening. So for some of the families I follow with this, we have a whole care plan where they know and everybody knows if that kid misses a night's sleep, we're on it and we're adjusting meds. In SHANK3 deletion, so Phelan-McDermott syndrome, they are more vulnerable to bipolar type presentations in adolescence. And there are some case level descriptions of inflammatory brain conditions related to skill loss. And so you may go more of a medical path with kids with regression in that setting to rule out some of these scary and treatable things. You heard for sure about connection to rare disease community. When enough clinical data is available and published, you can start to get literature reviews on your rare genetic condition. And if it gets to one called gene reviews, this is open access. Any doctor can Google it quickly, and it's super accessible and clear about what's known and what needs to happen. To me, that's moving the needle towards understanding for your condition. And so this is kind of the work we're all tasked to do. I'll also say growth is a place where rare genetic diagnosis I found very helpful. For some rare genetic syndromes, we know their growth curves are different. And that's what I'm showing here for Cornelia de Lange. Many kids who are later diagnosed with rare genetic conditions get labeled with things like failure to thrive when they're little. This can be really hard and scary for everyone because you're trying to feed a baby who doesn't want to eat. And in hindsight, it may be that they were actually just following their normal curve for their condition. And so this is helpful when they're little, but also when they're growing up and we're doing things with medications that might affect growth. I'm going to go 1 minute over, or 2, I'm sorry. So precision therapy. So you're going to hear, you've heard a lot about this precision health concept. Right now, the reality is that these precision genetic therapies are not happening commonly. I'm talking maybe 30 to 100 examples of either RNA or DNA-based therapies in the brain space, either ongoing or concluding for these kind of N of ones, but it's coming and it's happening more and more. I would say I'm working a little bit with a group called NLORM, and they do this in a charity way. And around 100 examples they have been involved with a precise molecule for an individual kid with a rare genetic condition. These are not benign treatments. So these tend to be kids with often degenerative or life-limiting courses. And they are interventions that come with high risk, but we are seeing it more and more. It's not going to be the standard for everyone anytime soon, but some of our data suggests about 5% of these rare genetic conditions may have a mechanism that could be theoretically targeted. And so the work of the field over the next 100 years is to try and figure out how to do this safely and effectively. I will say in certain conditions, and I've listed some here, this is beginning to become a reality. And for example, AADC deficiency or spinal muscular atrophy, these precision genetic therapies are now approved and standard of care. So it's coming slowly for a small minority of kids. So that's future. A big win, I would say, for Ontario, and I'm proud to present this work, has been access to this genetic testing. So many of you may have had sequencing or a genetic result through research, but historically, getting access to genome-wide sequencing is hard to come by. You may not be eligible. You may need to wait a couple years to see a geneticist. And if you live in a rural part of the province, that may not happen. We put together a proposal with the Ministry of Health to make this more accessible and done in-house in Ontario, and this was approved. And so as of January 1st, 2025, kids in Ontario with a diagnosis of moderate to severe intellectual disability or autism with additional medical factors can now access, ordered by a pediatrician, genome-wide sequencing for genetic differences. So this is a huge step forward when it comes to getting this done. Yeah. And I would say largely based on the data from this group and the network to show the importance of understanding biology and impacting care. So next steps, I'll finish on this, I'm almost done. We are looking to fill these evidence gaps. So one thing we're doing is pulling together OBI data, so POND, but also CPNet, the cerebral palsy study, where rare genetic diagnoses are not uncommon. And we are leveraging this data with our health system data to understand what are the needs, what are the gaps, and are these kids even identifiable in health system data? Doctors and medical records are beholden to something called ICD-10 codes. So if you want to like see somebody and diagnose them, you have to go through this library, and most of them don't include rare genetics. So there are a number of initiatives now where we're looking to do that better so we can figure out who these individuals are, what do they need, and how do we direct services in the right way. So there are a couple projects with that ongoing. I will summarize. So advances in genomics have been huge. High impact rare genetic diagnoses currently guide care. Potential for precision therapies is growing, but definitely not a mainstream thing yet. It's coming. Glad Steve talked about small molecules because I didn't talk about that here. But our access to this testing is increasing. And that's a powerful thing because more and more people are going to be coming forward, identified, and really power is in numbers when it comes to changing systems. There are still big evidence gaps when it comes to what these diagnoses mean over the lifespan for individuals. And we need to be innovative and work together in order to fill those questions. Okay, I'll stop there. Thank you, sorry.
Okay, thank you, Steve, Danielle, and Mukti for your awesome presentations, for enlightening us. I always feel smarter after a panel like this. And I'm sure you're just amazed at what's happening in this province and in this country, and grateful for the fact that we're able to speak to the evidence, and that we need to continue to advocate for that in all the things that we do, that is combined with a lived experience story that comes together so beautifully through POND. So this is the time for some questions for our panelists. So if you have a question, please indicate who your question is directed to, or if it's for anyone who wishes to answer. And I'm gonna, for those of you online, I'll be checking my screen to see if there are some questions here as well, and we'll try to take turns. So I see a question right here, and you'll have to turn your mic on. I think I see the red button there.
Hello?
Yeah, there you go.
Someone who has the solid light.
So one turns off.
Am I first? Oh, sorry. My question is for Danielle. I wanted to refer back to the slide you showed about depression versus psychosis. And I'm just wondering whether or not whether you've looked into or found that there's a correlation between depression and autism and perhaps bullying in schools, given the age range you were looking at there?
Yeah, so I love that question. And we looked at it so there's a large amount of data talking about the harms of bullying. In the POND data, so this was only about 80 kids who we had over time data. This was a longitudinal analysis. Bullying was a factor, but when you put it all into a model altogether, it was loneliness stood out more so than bullying. And that really resonated for me because there are some neurodivergent individuals where they are socially not that connected, and that's okay. And that's not distressing to them. But it's this mismatch between, I wish I had a bigger social group, I wish I had more friends, and I don't. That seems to be a unique vulnerability factor that stood out even when we're accounting for other stuff in the model. So for this paper, I would say it's a small paper, it's a student paper, but it has shaped my care that for some teenagers, the intervention for depression is social connection and really the whole community trying to find a place for this kid to fit and flourish. So more so than a medical model.
Thank you.
Thank you, all the panelists, for their wonderful presentations. My question is to Steve, and it's about the next release date for the missing database. When do you plan to release the next set of data?
So the question is, when is the next data release? Coming soon to a theater near you. We're actually waiting to get some new data transferred in from other projects. So it'll be probably in the next 6-month period for sure.
Thank you.
And maybe I can also use that just to mention another question that I'm sure is going to come up. But so for roughly 20% of the families enrolled in the study, we find a rare genetic finding result, and you'll get that back. Once we get that finding, you'll hear from one of our genetic counselors. So we often hear, what about the other 80%? If you don't hear back from us, and it's been a long time, feel free to email us in the study. And we don't generate negative reports. It's just we don't have the resources to do that. But we can give you at least an update of where the study's at. I would say in the paper I mentioned, we actually returned a few results to a few families that were collected 25 years ago. So you saw in the one slide, Peter Zatmari was in the photograph from the McMaster. He was at McMaster then. Many of you trained here and probably were diagnosed by him. So it was one of the 6th family that he collected. We returned the result just in the last year or so. So sometimes it takes time. We're usually faster than that. But I would comment very quickly, though, the research, so it's really important what Danielle said. Now it's moving into the clinical testing. The research will always give you a higher resolution analysis because we're always testing AI algorithms and new experiments and things like this to look at the data. But moving into the clinical, it'll be very reliable on the turnaround. It should be within probably from taking a blood sample to within 2 months, I would say, right?
The target is 3 months. It's 6 right now, but they're trying to get back to 3.
Our report was very positive. We have a child with an ultra rare condition, the only one in the world. So
the only one?
I will ask Danielle some questions after.
Do you know the gene name?
Ilya Belkin, and my child name is Lucas.
But the name of the gene involved?
Oh, the gene MED12. I'm sorry.
MED12? Yeah, okay. Just push off and then go to the next question.
Okay, there was a couple of other hands I saw. I'm not sure who was first. The red light goes first. Okay. Yeah.
Cool. So hi, my question's for Mukti. So great presentation. Thank you so much. And I think it sort of jumps off there. I've been talking to Matthew Cantor. He has Meyer Syndrome Foundation. He couldn't make it here today. He actually didn't know this was happening. I told him it's happening. You should come here. But the question would be around various different groups doing ultra rare diseases. So obviously you have 200 people. I don't know how big the one Matthew has, and this gentleman over here says is the only child in the world. And so are you working with other? Is this an idea to try to strengthen numbers kind of thing to try to sort of help each other out?
Yes, thank you for your question. Yes, we do collaborate not only with researchers globally, but we also collaborate with other rare disease organizations. Yes, we do. We are right now in the process of a collaboration. I'm not allowed to speak much about it, but yes, we do have a a parent organization that approached us a couple of weeks ago, and it's in talks, and we hope that in future we'll have more collaborations that will enable better approach to research and expand the research, because Ashmonell is an ultra-rare genetic condition. We only have 5 in Canada right now, and some of them in the United States and South America and Europe. So because it's so dispersed, it's very difficult to have a dedicated research team. So we are always up and open for collaboration with researchers globally, as well as with other organizations that are far ahead of us and who can give us better inputs and insights. I hope I answered your question.
Thank you. Yeah, thank you.
Great. Thank you. And there's one question that was online, so I'm just going to ask that one and then we'll get to you because I think you were next. The question is, we had participated in POND at Sick Kids. Do we have access to new testing or new discoveries in gene mapping with POND?
The answer is yes. We're always testing new algorithms, new approaches, new data sets, and I think it's safe to claim we're the leading study in the world. So yes, absolutely.
Okay, for the folks online, I hope you were able to hear that and that came through clearly. Okay, there, yes.
No, we have the solid, sorry. First with the solid.
Can you hear me? Yeah. Hi, my name is Shabs. My question is to all the panelists, Mukti, Danielle, and Stephen. You guys talked right from partnership to like loneliness, which is one of the factors. And in this room, we have probably like 100 people around us. And how do we collaborate as a community, as a group, where we can all help each other? Some of us are parents, some are researchers. So, having an online directory or some kind of collaboration platform. The reason I mentioned is that I'm part of a group called Popin, which helps you connect digitally. Is this something of interest which would help the community here?
Can you say the name of your community again?
It's called Popin. It's a group which you can pop into multiple events. So this could be a Popin community,
I love that. I will say, so through POND, there's formal structure to this, right? We have family advisory, a participant advisory committee, and events like this. But the idea of scientist matchmaker services is coming up more and more. So I'm not aware of us doing that yet, but there's conversations about it. So Steve may know more. The idea of, a central meetup online place where you can form those individual synergies, I really like.
So we have a question at the end of the day that I would like, if you're still here, I'd like to come back to, because we're going to ask you to have a conversation at the end of the day about how to communicate with you and in what format. Yeah. So if you can hold on to that idea, we would love to talk much more about it. It's on our agenda for the day.
Yeah, I just had an amazing idea based on your question that, we focus mainly on, as you heard from Mukti, the families that have the same genetic gene involved, but we could look at the cellular pathway, get those families, that would increase our numbers of people. That'd be really kind of neat. Mm-hmm. So can you push your button off so the next person can get their priority? Yeah. Okay,
great. Actually, the gentleman in the gray shirt and the beard, you've been waiting for a while. I don't know if you still have a question. No, it's a, yeah.
My question was for you, actually. Yeah.
The host is posting.
Yeah, my question is just on genomic sequencing equipment manufacturers. If there's any new innovations that reduce the cost of genome sequencing to allow for more samples being sequenced.
Great question, and it actually comes back to the last question. I forgot to fully answer. So the technology that we're using for the POND study and for this Canadian Precision Health Initiative 100,000 I talked about is called the Illumina short read sequencing. You sequence every nucleotide in the genome roughly 30 times, and then you use computation to reassemble a genome picture, if you will. So that's a state of the art, and we have negotiated a price at the national level due to the volume of 100,000 genomes. So it's actually it's around it's $560 Canadian, so it's about 400 or so American dollars, which is just unbelievable when you think about it. Now there are newer technologies, coming back to your question, called long read genome sequencing. We have them here also. There's a company called PacBio, another one called Oxford Nanopore, and it allows you to read longer strands of DNA sequence, so it captures different types of genetic variation we might miss in the short read approach. So we're actually doing that. It comes back to the last question online, is we're testing families that we think are highly genetic, but we haven't found something using the conventional technology, i.e. the one that I presented data on. We're running those samples now, and the consent process we collected all of your data on covers that already. Okay, now the other kind of neat thing is the long-read genome sequencing allows you to look at methylation changes. So sometimes when your environment changes, the way your DNA sequences are read can be modified. And genes can come on or off. It's called DNA methylation, or epigenetics is a term you might read in the media. So we've actually attributed probably 1 to 2, maybe 3% more findings looking at the epigenetic changes in the DNA. So that's kind of the next advance. And ASH1L codes a protein that is involved in modifying the methylation profiles across the genome. So again, one step at a time, we kind of gather all the data together. But I really wanted to reemphasize yet again is we're using all of your DNA samples as a prototype to push the whole scientific field together. So kudos to all of you for participating.
Awesome. I think we have time for one more question. I see a flashing light here in the middle. I don't know if that is yours. Okay. It's okay.
Oh, I think
here's the last question. Yeah.
My question is related to the genetics of parents, because I wondered, would there be ever the thought to screen the parents and like have them clinically assessed for autism? And then parents who it's like, oh, you're finding out late in life you have autism. And then there could be a better understanding of that genetic underpinning.
Yeah, so that's a great question. And it does happen sometimes. So when you order a genome-wide sequencing, and as you likely would have done through this study, they do it in trios, so both parents and child, and that's to understand, is that genetic change, if let's say the child has the condition, but the parent doesn't, that's some evidence that it may be associated. But you're absolutely right, this scenario where parents also have traits, and sometimes undiagnosed. So clinically, we see that a lot where a kid has a diagnosis, and then the parent comes and is like, oh, wait, this explains a lot. And so the idea of bringing together parent phenotype and genotype with kid phenotype and genotype does happen in the rare genetic, when we're making reports about new disease genes. It so far is not a part of the POND protocol. We don't do parent phenotyping apart from history, but it is a methodological approach. I know Advocacy wants to build on it.
Well, I mean, we do a bit of it. So when we have a child that has first of all, the heritability doesn't stay true to diagnosis. So we need to be clear about that. We can have parents with ADHD traits who have a kid with autism or vice versa, right? Like the traits kind of cross across the neurodevelopmental condition. But what we do currently in POND is if the child has a variant and the kids, and then we test the parents to see if it's inherited or whether it's de novo. If it's inherited, you get a call from us. It's not a full assessment, but we do a bit of a history to collect the traits to help with the interpretation of the variant back, because Steve's team would need the information to say, is this variant in an interesting gene but has no impact really? Or is this a variant in an interesting gene and it seems to track with the generations? So we do collect information. We just don't do clinical diagnosis for parents. But I've been in a position multiple times where I'll collect the history for the purposes of interpretation and say, you want to visit your doctor.
Yeah, we had that scenario where it was like, oh, let's take, and oh, actually it's a variant, so it doesn't look like it came from me. But it's like,
can I just jump in? This is so important. It's an important question, but it's kind of built into our process, and it informs us in our studies. So if you had really good eyes and saw the table I pointed out of the first 100 who got their data back, about 20% were inherited variants. And we would then say, okay, let's contact the family and call them back in to see if they have any of these related traits. May not have the same diagnosis, but what else is going on? And almost always we'll also look at siblings at that point in time and do genetic testing on them too. So it's really, really important question.
Okay.
Mark.
Yeah.
Can we hear, if we have 1 minute, I want to hear just John's story.
Okay. 1 minute we've got. All right, go ahead, John.
I'll try to do it in 60 seconds. There's a lot to say.
Maybe just introduce yourself again, John, for a moment.
So my name is John Oldenhoff. I actually did my PhD at U of T. I have been in clinical research for, I guess, 25 years. I worked on 2 drugs that when my son was born and got a genetic epilepsy, he's actually on. And I've been, like Mukti, I've been on a journey of being part of a foundation to help drive therapies. And Mukti, you know, your presentation was great because that's exactly where I was, I guess, back in 2013. We made connection with 65 families. We now have over 1,000 families worldwide. And just 2 weeks ago, Capseda, and I can say this because it's public knowledge, has just gotten FDA approval to drive forward their gene therapy for SDXBP1. So I've kind of had a second postdoc in rare diseases. I've learned a lot in the way. I've had interactions with the FDA, just so many important things, and I'm happy to talk to anybody afterwards. But my main comment is, you know, POND is doing a great thing because they're taking an initiative. But for many rare diseases, parents have to take the initiative. There's not one rare disease group that is not parent-driven. And so get involved in whatever way you can. At the very least, make sure you let the foundations know that you're around. They have registries, super important to be part of it. And, you know, I guess maybe my other message just is I've been so impressed with the FDA. They have something they call patient-focused drug development. And they have actually, you know, I mean, a lot of these genetic diseases, we hope to hit the home run and cure it completely. But the FDA is very well aware that even small improvements will be a lifetime of help for you. And so when they talk to companies, they take this patient-focused drug development into account in terms of what is important and meaningful to parents. And, you know, we were talking to them about toiletry, and they said, well, it's not just getting toiletry. Even indicating that they need to go to the toilet could be an objective endpoint in a clinical study. So just important to get involved. And again, Mukti, I'd like to talk to you afterwards too, but just in general, US parents are the ones that are going to drive things. Also the clinical genetics, so important, right? Companies don't start paying attention to a disorder until it's identified. And if nobody identifies it, it's, you know, you don't get an interest. We were originally considered 1 in 100,000. We made it to 1 in 30,000. And all of a sudden, all these companies started becoming more interested because they knew that there was more of a chance for them to complete the studies, but then in the end also to be able to sell their therapies. So thanks.
Thanks so much, John. As Peter's coming up, I'm going to invite you to join to speak. Can't thank all of our panelists, Steve, Danielle, and Mukti.
Thank you, Marg and panel, for a really interesting session. So we're heading up on our lunch break now. We're running a little bit behind. One of the things that we've asked, the organizers have asked, is when you leave the auditorium, just in front of the registration area, there's a whole series of steps down the way. They're asking if everybody can have a seat there for a group picture of all the participants. So obviously, if you're not interested, go on by, but it'd be nice to have a record of everyone. Also, we'll have the fair going. I just wonder if the folks who are doing some of the fairs could just stand up for a quick moment. Okay, so if you look around the room, these folks are going to be running various booths all around the space. Please go up and talk to them. And one thing I'm going to remind you of as well, and sort of part of the art of conferencing is, we're all here, shared experiences and stuff, is introduce yourself to some other folks. Sit at a table, ask each other about your kids. You might find somebody new who's gone through an experience you haven't, can give you some insights on stuff, because this is a room full of experts. And Dr. Scherer will be meeting the families at the registration table for the tour at 12 and 12:30. And at 11:50 for the folks that are joining us virtual, I think we're going to be starting to do our virtual tour of some of the fair booths for the folks to see along the way. So I think, are we regrouping at 1 or are we going to go a little later?
Regroup at 1.
Okay, so we're going to regroup at 1. Warning, I've deputized my friend over there, Adam, to be a fellow people wrangler, so you don't want to face his wrath. Get back on time. Okay, everybody, out for the photo. It's time for your close-up.